Okay, this is Mr. Maxwell's AP Biology class presentation on breathing and respiration. So this is intended normally to be an interactive classroom discussion, but I'm going to present it here and I'll leave places for you to pause so that you can think about the questions. Remember, if you think about stuff and try to come up with it, you'll remember it better. So, first question is, how long can you hold your breath? And so what you ought to do is pause this and see how long you can hold your breath. Okay, so why can't you hold it any longer than that? And what do you need air for anyways? These are questions I'd like you to stop and think about for a couple seconds before we go on. In class we'd have a discussion here, but so think about it for a while. Okay, so hopefully you came up with the fact that your mitochondria need oxygen in order to be able to do cellular respiration. Because without cellular respiration, you don't make any ATP. If you don't make any ATP, your muscles don't contract. Your muscles don't contract, your heart doesn't pump. Your heart doesn't pump, your cells don't get any oxygen, and you're out of luck. So, the oxygen is absorbed from a capillary bed, and the carbon dioxide's dumped off into a capillary bed. And of course, this oxygen comes through arteries from the lungs, and it's across the lungs where you get oxygen dissolving into the bloodstream. And of course, in us, the respiratory surface is the lungs. It could be the skin of some animals, and for us, the respiratory medium is going to be air, of course, but some organisms like fish, it's going to be water. So this diagram shows the difference between cellular respiration and physiological respiration. Physiological respiration really is just ventilation plus the dissolving, and then cellular respiration, of course, is the metabolic stuff that happens in the cell and the mitochondria. So a little point here about the grammar of respiration. The word respire is a verb, and it goes, it respired, it is respiring, it will respire. Not it respirated. Never use that. Okay, so I'd like to take a look at these gills of this mud puppy here, and see if you can connect form and function. These red things sticking out the side of the mud puppy are its gills. So hopefully you came up with the idea that having this feather-like filamentous stuff floating out into the water increases surface area to volume ratio, and of course that allows it to do more oxygen uptake and carbon dioxide transfer. So as a general rule, anytime you see surface area, anytime you see a folded structure like this, you should immediately think surface area to volume ratio, because that's what it's always for. Okay, now fish have gills too, but they typically have them inside the body, underneath an operculum. The operculum is a bony plate here, and so they breathe water in, pass it over their gills, and then the water comes out through the gill slit, and that's how they ventilate their gills. So one question you might ask is, which type of flow is going to be better at getting oxygen out of the water? One way of doing this is to arrange it so that the blood and the water go in the same direction, and that's what we call concurrent flow. The other way to do it is to have the blood and water going in opposite directions, and that is called countercurrent flow. So which type of flow is more efficient? Stop and think about it for a couple seconds, and then I'll talk about it. Okay, so in the top diagram here we have countercurrent flow. In the top diagram we'll talk about this being the water flowing across the gill here, and it's going to start at 100% oxygen saturation, and then this on top here we're going to think of as the blood's coming from the tissues, and it has 0% oxygen because it's all been used up, and it's going to flow this direction. So at the beginning of this there's a very steep gradient for oxygen to diffuse into the blood. As you go further down along this, you get to a point where the concentration of oxygen in both water sources is the same. In other words, near 50%. And so 50% is going to be about the maximum oxygen you're going to be able to extract out of a concurrent flow situation. If you compare that to a countercurrent flow, the bottom diagram here is countercurrent flow, and we start with the top here being oxygenated water entering the gill, and the bottom here being deoxygenated blood returning from the tissues. As the water flows over the gill, it's encountering blood that has less and less oxygen in it. And so even at the far end here, where the blood might be, where the water might be down to, say, 10% oxygen, it's still a downhill or a down gradient, and the oxygen still transfers across the membrane into the blood. And so when we get up here where the blood's really, really oxygenated, we have fresh water coming in from the outside, and it's as oxygenated as it's going to be. So clearly countercurrent flow ought to be the more efficient. And if you look in fish, you'll find that that's exactly the way they do it. Blood flow will be over the gills in one direction, and water flow will be the opposite direction of that. And you can imagine that natural selection would, of course, select fish that had blood flowing the right direction that would get the most oxygen out of the water. And here's another diagram that shows it. If you don't understand what I was saying in the other diagram, examine this diagram a little bit, and I think you'll see that countercurrent flow works real well. Okay, so now if you live in air, you have to first take in oxygen from the external environment, have it dissolve across a membrane, and then get into the bloodstream that way. So all respiratory surfaces in animals that live terrestrially are going to have some moisture associated with them. And let's just look at how frogs breathe. I think this is a really cool video. You'll just watch this for a couple seconds and you'll notice a couple things. They're moving. He's got a little nostril flap that keeps shutting. He's moving his throat here, and he's moving his abdomen here. And so basically what happens is when he lowers his mouth here, air gets sucked in through the nostril, and then he closes his nostril and pushes that air down into his lungs. And so this is why it's called positive pressure breathing, because you're using positive pressure to push the air. Then he's going to contract his abdominal area here, which pushes it out into the mouth, and then he opens his little nostril flap here, breathes it out, and can take another breath. So contrast that with us. We do negative pressure breathing. We have this diaphragm here, and hopefully this is a review from like health class or 6th grade biology or something. But you know that when the diaphragm contracts, it gets shorter, and the shortest distance between 2 points is a straight line. And by contracting this, it makes the cavity here bigger, which means that you have a negative pressure and the air gets sucked into the lungs. Okay, so building on that, there are some things you ought to know about. Terms you ought to take down are vital capacity, tidal volume, and residual volume. And I'd also like you to think about whether, when we're done looking at this, if it would be possible to design a more efficient lung. Okay, so when your regular breathing is just this part here, and that's called your tidal volume. When you breathe in really deeply, that's the maximum you can breathe in. That's your inspiratory capacity. And then if you breathe it all the way out, that's your expiratory reserve volume. So anything below where you normally would breathe out. So that's what that is. the amount of air from your maximum exhalation to your maximum inhalation is called your vital capacity. And it turns out that we actually have a little bit more air in our lungs than even we can breathe out at our maximum amount. And that's called your residual volume. And you really need that residual volume to keep the sides of the alveoli from kind of sticking together and for your lungs from collapsing. But the downside of this is that makes it be dead space where you're going to have a high concentration of carbon dioxide, and any new air coming in is going to mix with that, and it immediately reduces the amount of oxygen in that air. So if there were a way to get rid of the residual volume, you'd have a more efficient lung. And if you look in nature, it turns out that you can find something that has that, and you would be looking in birds, because birds, of course, fly and can fly long distances, and they have a really cool one-way lung. Basically, the way they breathe is in their first inhalation, their breath, the air comes and gets stored in these posterior air sacs. When they breathe out, then, the air sacs empty into the lungs. And this lung is one way because in their next in-breath, they export the lung volume into these anterior air sacs. And then when they breathe out again, the anterior sacs are emptied through the trachea and out the mouth. And so the air only goes one way through these lungs, and you could probably guess that the capillaries are going to be oriented such that they're in a countercurrent fashion, just like in a fish's lungs, and of course they are. So this lung is really cool because it eliminates that dead volume, and it also uses countercurrent flow to maximize the absorption of oxygen. So I want a bird lung transplanted into me. Okay, and this diagram pretty much shows exactly what I told you. You got your first breath comes into the posterior air sac, you exhale into the lung, which is a one-way lung, then you inhale, and that puts it in the anterior air sacs, exhale, puts it out your mouth, if you're a bird, of course. Okay, so you should at this point be able to compare and contrast the respiratory mechanisms in these four animals, and at this point I would suggest that you pause and go ahead and do that.